Chapter 14 of your textbook covers molecular oncology. The objectives covered in this chapter include contrasting tissue-specific and tumor-specific molecular targets, listing molecular targets that are useful for diagnosis and the monitoring of solid tumors, explaining how microsatellite instability is detected, showing how clonality is detected using antibody and T-cell receptor gene arrangements, and describing translocations associated with hematological malignancies that can be used for molecular testing. Cancer is caused by non-lethal genetic mutations affecting certain genes. Proto-oncogenes are normal genes which, when altered by mutation, become an oncogene that can contribute to cancer. Proto-oncogenes may have different functions in the cell. Some proto-oncogenes provide signals that lead to cell division. Others regulate programmed cell death. Oncogene mutations are usually a gain of function and a dominant mutation. Tumor suppressors are genes that normally arrest cell division. Mutations in tumor suppressor genes are usually a loss of function and are recessive mutations. As mentioned before, cancer is caused by non-lethal genetic mutations. Normally, cells grow in the presence of nutrients and factors that stimulate cell division, known as mitogens. Lack of mitogen stimulation results in cell arrest or apoptosis. If oncogene or tumor suppressor gene mutations stimulate aberrant growth signals, cells grow in the absence of controlled stimulation. Although not yet completely standardized, several tests are performed in almost every molecular pathology laboratory. These include tests that target various markers, such as tissue-specific markers, disease-specific markers, and viruses. Tissue-specific markers are molecular characteristics of the type of tissue from which a tumor arose. They can or are also expressed on normal cells, so the presence does not always prove the presence of a cancer. Disease-specific markers, such as tumor-specific genes, are not expressed on normal cells and are more definitive with respect to the presence of a tumor. Specific targets include translocations, point mutations, and polymorphisms in tumor suppressor or oncogenes. Viruses are similar targets to the disease-specific targets, in which they have a more definitive result indicating infection with the virus has occurred. Methods for detection of the various diseases include hybridization, the different blotting methodologies, standard PCR, RT-PCR, electrophoresis, PCR with heteroduplex analysis, SSCP, and real-time PCR with gene or patient-specific probes. The gene and chromosome abnormalities observed in cancer include gene mutations such as oncogenes and tumor suppressor gene mutations, chromosome structural abnormalities such as translocations, deletions, and insertions, and chromosome number abnormalities. Examples include aneuploidy and polysomy. Various tests are routinely performed to aid in the diagnosis, characterization, and monitoring of solid tumors. Some have been part of molecular pathology for many years, while others are relatively new. The methods applied to detect molecular characteristics of tumors are described in the next section of this chapter. The first molecular abnormality in solid tumors is the HER2/neu gene. In normal cells, the protein coded by the HER2/neu gene is required for cells to grow and divide. It's overexpressed in 25 to 30% of human breast cancers, and this is a predictor of more aggressive growth and metastasis of the tumor cells. It's also an indication for use of the anti-HER2/neu antibody drug Herceptin. This therapy works best on overexpressing tumors. Therapy is indicated for HER2/neu positive women with breast cancer that has spread to the lymph nodes or other organs. Testing for the HER2/neu abnormalities is often performed by IHC, immunohistochemistry testing. It uses monoclonal and polyclonal antibodies to detect the HER2-neu protein, and then is observed microscopically. FISH, which measures DNA and RNA of HER2-neu, is more reliable than IHC, especially in older fixed tissue samples. The ERBB family of growth factor receptors include the HER2 receptor and EGFR. The EGFR gene family stands for the human epidermal growth factor receptor. The factors that bind to these receptors on the cell surface begin with a cascade of events, including autophosphorylation and phosphorylation of the other proteins by the receptors. These factors include the epidermal growth factor, EGF, human transforming growth factor alpha, or TGF-alpha, heargulins and neargulins. EGF and the NRGs, or neargulins, are small peptides that are active in the development of various cell types, such as gastric mucosa, the heart, and the nervous system. This diagram depicts the EGFR gene family. The epidermal growth factor receptor oncogene encodes another of the same family of epidermal growth factor receptors. This gene is mutated or amplified in several types of cancer cells. Tumors with activating mutations in the EGFR are sensitive to tyrosine kinase inhibitors. TKIs such as Tarceva, Iressa, and Sutent have been studied in clinical trials. EGFR protein is detected by immunohistochemistry staining. The EGFR gene and chromosome abnormalities can be detected by FISH. And the EGFR gene mutations are detected by SSCP, qPCR, or direct sequencing. Detection of mutations in the EGFR kinase region is now also performed to guide treatment strategy using EGFR inhibitors. The Kirsten rat sarcoma viral oncogene, known as KRAS, encodes a key component of cell signaling. Mutations in KRAS are the most common oncogene mutation in cancer. These mutations are associated with tumor malignancy and may also affect response to some therapies. KRAS gene mutations can be detected by single-strand conformation polymorphism or direct sequencing. The 53 kilodalton tumor suppressor gene, known as TP53, encodes for transcription factor. This gene is mutated in half of all types of cancer. Loss of the TP53 function is an indicator of poor prognosis in colon, lung, breast, and other cancers. Mutations in the p53 protein can be detected by immunohistochemistry testing. The TP53 gene mutation itself can be detected by a variety of methods, including SSCP and direct sequencing. The textbook goes over in more detail various genes associated with solid tumors, but we won't be going into them in as much detail, but they're listed here. So there's the Ewing sarcoma, EWS gene, the synovial sarcoma translocation at chromosome 18, and the synovial sarcoma breakpoint 1 and 2, the paired box forkhead gene in rhabdomyosarcoma, the ataxia telangiectasia mutated gene, the von Hippel-Lindau gene, the V-MYC avian myelocytomatosis viral-related oncogene, that's neuroblastoma-derived, and the rearranged during transfection proto-oncogene. Table 14.4 in your textbook provides a good summary of the molecular abnormalities in some solid tumors. It includes the associated gene, the location, the mutation, detection methods, and then associated diseases. Once a gene mutation is identified in a family, targeted resequencing is used to test for the mutation in other family members. In an inherited condition, such as inherited breast and ovarian cancer, blood samples are sufficient for mutation analysis in unaffected individuals. In tumor cells, further testing for loss of heterozygosity may be performed. Loss of heterozygosity reveals the loss of the good allele at a locus, uncovering the homologous locus with a recessive mutation. Inherited tumor suppressor gene mutations are recessive for the malignant phenotype, but the tumor suppressor gene mutations are dominant with respect to increased risk of malignancy in an individual. Loss of heterozygosity and increased risk of malignancy can be explained by the two-hit hypothesis. Most genes require two mutations to cause a phenotypic change. In the top example, you have a normal individual with no mutations, an at-risk individual with a mutation located on one area of the chromosome, and then an affected individual with mutations on both genes. Dr. Alfred Knudsen developed a two-hit hypothesis by statistical methods without any experimental approaches. He was comparing the time at which both eyes would be affected by retinoblastoma and eye disease if one or two hits were required, and then predicted the relative chance of this occurring. Knudsen's hypothesis postulated the recessive nature of tumor-initiating gene mutations and the mode of inheritance in familial cancer. This led to the concept of the existence of tumor suppressor genes and loss of heterozygosity as being relevant to carcinogenesis. In the bottom advanced example, you have the at-risk individual with an inherited mutation. The affected individuals can then be persons with a mutation on both, or a person who experiences loss of heterozygosity, in which the loss of the good allele at a locus uncovers the homologous locus with a recessive mutation. Loss of heterozygosity is detected by PCR and capillary electrophoresis of heterozygous short tandem repeat loci, also known as STR. These are linked to the disease genes. A deletion or loss of the normal allele uncovering a recessive mutant allele is identified by the loss of the short tandem repeats. In the example below, loss of a linked heterozygous STR implicates a concurrent loss of one gene allele. So on the top you have the normal with the heterozygous STR, so you have a normal allele and a mutant allele. And on the bottom is showing a tumor in which the normal allele appears shortened and the tumor allele is apparent. BRCA1 and BRCA2 are tumor suppressor genes associated with inherited breast cancer risk. They encode proteins that participate in DNA repair. Inherited mutations in these genes significantly increase risk of breast cancer at an early age. The frequently occurring mutations include the 187 deleted AG and the 538-2 insert C in the BRCA1 gene, and the 617 deletion of T in the BRCA2, and these can be detected by qPCR and other methods. Most mutations can be detected by direct sequencing of both genes. This figure shows an example of direct sequencing by sequence-specific PCR in which the known location of the mutation is targeted with a mutation-specific primer. In this example, the 180 base pair fragment that can be seen here lightly between the 230 and the 120 base pair indicates the presence of the mutation. This is seen in lanes 3 and 4, the mutant lanes of the agarose gel. Lynch syndrome, or hereditary non-polyposis colorectal carcinoma, is an inherited form of colon carcinoma, accounting for 5% of all colon cancers. Predisposition to cancer in this syndrome is caused by mutation in the MSH2 and MLH1 genes. Mutations have also been found in the MHH6 and HPMS2 genes. These genes are responsible for correcting replicative errors and mismatch bases in DNA. Replication repeat errors, which are caused by slippage between the replication apparatus and the DNA template, and mismatch bases in DNA, are normally corrected by the mismatch repair system, or the MMR. This process is encoded by the MLH1 and MSH2 genes along with several other genes. Replication errors occur especially in microsatellites, which are short tandem repeats where 1 to 3 nucleotides are repeated in the DNA sequence. Microsatellites are especially sensitive to errors during DNA replication. Microsatellite instability is associated with the mutations in the MSH2 and MLH1 genes, along with the other genes involved in the repair process. Microsatellite instability is the production of new alleles from unrepaired replication errors. As mentioned before, the replication errors result from the slippage during DNA replication. If the error is not repaired, the next round of replication will create a new allele of the original locus. Additional uncorrected errors will produce more alleles. 85 to 90% of the hereditary non-polypopsis colorectal cancer tumors have microsatellite instability. Mutations in genes of the mismatch repair system, or the MMR, are inferred by testing for microsatellite instability. MSI analysis determines gene function, and direct sequencing is used to detect the actual gene mutation. The National Cancer Institute has recommended that the screening of 2 mononucleotide repeat loci, BAT25 and BAT26, and 3 dinucleotide repeat loci, D5S346, D2S123, and D17S250 is sufficient for determination of MSI, microsatellite instability. Alternative markers have been proposed, and some laboratories test additional loci to ensure amplification of at least 5 loci. In addition, mononucleotide repeat structures may be more sensitive markers for microsatellite instability than the dinucleotide repeats, so some laboratories prefer mononucleotide repeat loci instead. Microsatellite instability is detected by comparing PCR amplicons of the microsatellite loci. Unstable loci appears extra products in the tumor tissue compared to normal tissue. Microsatellite instability as detected by gel electrophoresis results can be seen in this figure on the left. You can observe the increased tumor alleles designated by the T lanes compared with the normal cells designated by the N lanes. Stability is indicated by identical patterns in both tumor and normal cells. An example of this is the normal and tumor cells in the middle lane. On the right is an example of microsatellite instability detected by capillary gel electrophoresis. DNA from the tumor, again designated by T, is compared with the DNA from the normal cells from the same patient designated as N. Increased alleles in the tumor scans reveal instability at those loci, so the example showing the top 4 scans, and stable loci look the same in normal and tumor tissue, the bottom 2 scans. So up to this point, we've discussed gene and chromosomal mutations in solid tumors and the molecular detection techniques for those types of cancers, along with microsatellite instability. The next group of slides will be discussing molecular detection and associated genes with leukemia and lymphomas. Targets associated with hematological malignancies include antibodies, gene arrangements, translocations, point mutations, polymorphisms, and viruses. The methods for molecular detection include hybridization, blotting methodologies, standard PCR, real-time PCR, electrophoresis, PCR with heteroduplex analysis, SSCP, real-time PCR with gene or patient-specific probes. So a lot of these methods are similar to what we saw with the detection of solid tumors. Gene rearrangements analyzed for hematological malignancies include VDJ recombination, the normal intrachromosomal rearrangements in B and T lymphocytes, as well as the abnormal interchromosomal translocations that can occur in any cell type. Gene rearrangements are normal events that occur in lymphocytes. Antibody genes, such as immunoglobulin heavy chain genes, immunoglobulin light chain genes, and T-cell receptor genes can rearrange. Gene rearrangement occurs independently in each cell. As mentioned earlier, gene arrangements are normal processes that occur in B and T lymphocytes as they mature from lymphoid stem cells. The genes coding for immunoglobulin heavy and light chains begin with the rearrangement process in early B cells and in pre-B cells. The T-cell receptor genes rearrange in the order of gamma, beta, and alpha chains, and then differentiate into the cytotoxic T and the helper T cells. Remembering way back in immunology, each antibody consists of 2 heavy chains and 2 light chains. The locus coding for the immunoglobulin heavy chain is located on chromosome 14. The unrearranged or germline configuration of the immunoglobulin heavy chain locus consists of a series of gene segments or repeated exons coding for the functional parts of the antibody protein. The immunoglobulin heavy chain gene consists of variable indicated by V, diversity indicated by D, and joining indicated by J gene segments, and this is the germline configuration. The V segments are accompanied by a short leader region, L, coding for amino acids that will direct secretion or localization of the protein. One of each type of segment, V, D, and J, is selected and combined by an intrachromosomal recombination event. First D and J, and then V and D. The C, which is the constant segments, are joined through splicing or a secondary recombination event, known as class switching. Knowing that these gene rearrangements occur may be used to detect leukemias and lymphomas arising from cells that have rearranged their immunoglobulin or T-cell receptor genes. Gene rearrangements occur independently in each lymphocyte so that a normal population of lymphocytes is polyclonal with respect to their rearranged immunoglobulin or T-cell receptor genes. Overrepresentation of a single rearrangement in a specimen cell population can be an indication and characteristic of a lymphoma or leukemia. When over 1% of cells makes the same gene rearrangement, the cell population is referred to as monoclonal with respect to the rearranged genes. Clonality can be detected by Southern blot. Detection of clonality by Southern blot is described using the immunoglobulin heavy chain as an example. Restriction sites have been mapped in the germline configuration of the immunoglobulin heavy chain gene on chromosome 14. The enzymes BAMH1, ECO-R1, and HIND3 are commonly used for this procedure. When the germline sequence is rearranged, the restriction sites are moved, created, or deleted, resulting in a unique restriction pattern for every gene arrangement. DNA isolated from white blood cells is cut with the restriction enzymes, transferred to a nitrocellulose membrane, and hybridized with a probe to the joining region of the IgH gene. Normal results reveal the expected fragments generated from the germline DNA. When examining the autoradiogram, the normal fragments are visible for two reasons. First, a normal patient specimen contains cells other than lymphocytes that do not undergo gene rearrangement. The second reason for the presence of the germline bands is that only one chromosome in a lymphocyte undergoes gene rearrangement, leaving the homologous chromosome in the germline state. If the first rearrangement fails or is unproductive, then the second chromosome will rearrange, an event that occurs in fewer than 10% of lymphocytes for the heavy chain gene. Also, in a normal specimen, there will be millions of immunoglobulin gene rearrangements, so no one rearrangement is present in high enough amounts to be visible as non-germline bands on the membrane or autoradiogram. Thus, only the germline bands will be visible. If, however, 2 to 5% of the cells in the specimen consist of a clone of cells, all with the same gene rearrangement, the clone will be detected by the presence of additional bands different from the germline bands. The number and location of these bands is not predictable, as they depend on the nature of the gene rearrangement in the cell giving rise to the clonal population. They are, however, characteristic of that particular monoclonal population. Interpretation of the results are positive if bands other than germline are present, and negative if only the germline bands are present. This figure shows immunoglobulin heavy chain gene rearrangement by PCR with amplification from the variable region. Forward primers complementary to the variable region and reverse primers complementary to the joining region are used to amplify the diversity region, which is on top. In a polyclonal specimen, amplification products in a range of sizes will result. These products produce a dispersed pattern on an ethidium bromide stain agarose gel. In this figure, lanes 4, 8, and 11 show a normal population that yields the dispersed bands. If at least 1% of the sample is representative of a monoclonal gene rearrangement, that product will be amplified preferentially and revealed as a sharp band by gel electrophoresis. So in this example, you can see lanes 3, 6, 9, and 10 show the sharp band that indicates a monoclonal gene rearrangement. There are numerous chromosomal abnormalities associated with leukemias and lymphomas. Translocations can be used in diagnosis and monitoring of hematological tumors. Table 14.7 in your textbook provides a more comprehensive list of diseases and the chromosomal mutations associated with them. Some of the diseases you'll see in the table, although this list is not all-inclusive, include the pre-B acute lymphoblastic leukemia, the B-cell leukemia, acute T lymphocytic leukemia, acute myeloid leukemia, acute promyelocytic leukemia, acute myelomonocytic leukemia, chronic myelogenous leukemia, acute lymphocytic leukemia, Burkitt's lymphoma, diffuse large B-cell lymphoma, T-cell lymphoma, follicular lymphoma, mantle cell lymphoma, and multiple myeloma. Please make sure you take a look at Table 14.17 in your textbook to see the more commonly associated chromosomal mutations. Translocations and other abnormalities in chromosome structure and number can be detected by fluorescent in situ hybridization, or FISH. In this example, the translocation between location 8 and 14 on the chromosomes are detected using breakpoint methodology. Two probes labeled with biotin or digioxygenin are complementary to sequences flanking the chromosome 14 breakpoint. In the absence of the translocation, as seen on the image in the left, the probes will appear next to one another in the nucleus. The translocation shown on the right will move one probe to the chromosome 14, leaving the other behind on chromosome 8, and this results in separation of the signals in the nucleus. There are a couple important notes to remember when determining molecular methodologies to detect translocations used in diagnosis and monitoring of hematological malignancies. Translocations are detected with higher sensitivity using PCR, and quantitative PCR may be used to quantify tumor load during patient monitoring. Fluorescent in situ hybridization is recommended for initial diagnosis, whereas the PCR methodology is better for monitoring of patients' progress. It's important to remember that gene rearrangements in immunoglobulin and T-cell receptor genes are normal and take place regardless of the presence of malignancy. The unique nature of gene rearrangement systems with cell-specific antibody and antibody receptor formation are exploited to find abnormal cell populations clonally derived from these cell-specific features. Targeting these gene rearrangements, however, can only be applied to tumors arising from lymphocytes. Other types of malignancies, such as myeloid tumors, cannot be analyzed using these targets. Often, development of the cancer state results in DNA anomalies, translocations, or other types of mutations that can be applied to clinical discovery and monitoring of tumors. These abnormal genetic events aid in diagnosis of specific types of hematological tumors, such as translocations that are highly associated with chronic myelogenous leukemia, promyelocytic leukemia, or follicular lymphoma. These mutations usually involve tumor suppressor genes and oncogenes, and this has helped to determine the molecular events leading to the tumor phenotype. Several translocations are frequently found in certain types of tumors, and we'll discuss these on the next two slides. The translocation of 14 and 18 locations on the chromosome is a reciprocal translocation between the long arms of chromosomes 14 and 18, and this is found in 90% of follicular lymphoma cases and 20 to 30% of large cell lymphomas. With translocation, the B-cell leukemia and lymphoma, the BCL2 gene, is moved from chromosome 18 to chromosome 14. BCL2 is dysregulated and overexpressed when moved to chromosome 14. In this figure, PCR is used to detect the translocation of 14 and 18. The forward primer hybridizes to chromosome 18, while the reverse primer hybridizes to chromosome 14. Because the forward primers are only on chromosome 18 and the reverse are on 14, a PCR product will be generated only from the translocation 14-18 chromosome. Once the fragments are resolved on agarose gel electrophoresis, you can see the molecular weight marker in the first lane compared to the positive specimens, along with the negative specimens in the middle lanes. And then the last lanes are the positive control, the sensitivity control, and the negative control. So the band size is determined by the chromosomal breakpoints. The T922 translocation is a reciprocal translocation between the long arm of chromosomes 9 and 22 and is found in chronic myelogenous leukemia and acute lymphoblastic leukemia. This translocation forms a chimeric gene between the breakpoint cluster region, the BCR gene, on chromosome 22, and the Absalom leukemia virus, or the ABL gene, on chromosome 9. The translocated chromosome is known as the Philadelphia chromosome. The chimeric gene BCR-ABL produces an abnormal protein that drives the tumor cell phenotype. P210 BCR-ABL and P190 are the two main fusion proteins produced by the T922 translocation. They differ in the amount of the BCR gene that is attached to the cABL gene. The T922 translocation begins with breakage of chromosome 9 and 22 in the introns of the BCR and c-ABL genes, which are here. The breakpoints are located within the introns of both genes. The same rationale described for the T1418 translation detection by PCR is used for the translocation of 9 and 22 chromosomes. That is, the forward primers are designed to hybridize to the BCR gene on chromosome 22 and reverse primers to chromosome 9 in the c-ABL gene. A product will result only if the two genes are joined by the translocation. Unlike the BCL2 gene translocation breakpoint, which is 3' to the intact genes, the breakpoints in the BCR and ABL genes are within the genes, particularly in introns. The primer binding sites are located in the exons closest to the breakpoints of the genes. Due to the length of the introns that separate the primer's binding site, reverse transcriptase PCR has been commonly used. This figure shows results of a standard reverse transcriptase PCR test for the T922 translocation. Lane 1 shows the molecular weight standard, and then lanes 2 through 5 are positive patients for the translocation. Lane 6 is the negative control, lanes 7 through 11 are amplification controls, and lane 12 is the blank. The band size for the patients is determined by the different chromosome 22 breakpoints. The last molecular methodology we'll discuss for detection and diagnosis of hematological malignancies is quantitative PCR. Quantification by real-time PCR provides an estimation of treatment response. Although cytogenetic methods, especially FISH and standard reverse transcriptase PCR, are most practical for diagnosis and also in the early stages of treatment, quantitative PCR provides a valuable estimation of tumor load over the course of treatment. Several commercial reagent sets are available to detect translocations as the T922, the Philadelphia chromosome detection by quantitative reverse transcriptase PCR. Because transcript levels are being measured using quantitative reverse transcriptase PCR, it is important to stabilize the specimen RNA on receipt. Another recommendation is to collect at least 10 milliliters of peripheral blood for analysis to avoid false negatives. A standard curve or a high and low positive control and a negative control should accompany each run. Frequently used TaqMan methods report measurements as a ratio of the BCR-ABL transcript level to the RNA integrity control. The standard curve uses tumor cells diluted into normal cells. The standard curve that should be used should have transcripts of known copy numbers diluted into normal RNA. The figures on the left are actually depicting translocation 1418 analysis by real-time PCR. The standard curve is established using cultured cells with the T1318 translocation, counted and diluted into cultured cells without the translocation. The results shown are from DNA isolated from each mixture of the cells and analyzed by real-time PCR using a TaqMan probe. So, in summary, we covered in this chapter that molecular testing analyzes tissue-specific and tumor-specific targets. Genome, chromosome, and gene mutations are useful targets for diagnosis and detection of solid tumors. Microsatellite instability is a test for function of the DNA mismatch repair system, which may be mutated in hereditary colon cancer. Immunoglobulin and T-cell receptor gene rearrangements are tissue-specific markers for certain lymphomas and leukemias. And translocations are tumor-specific markers for some hematological disorders and malignancies.